Okay. Lastly, let me just briefly talk about the cell-mediated immunity. Although we've talked about the cell-mediated immunity in details, generally we've touched on the cell-mediated immunity. So it's kind of a repetition. So it is majorly run by T cells. We know that. We say there are two types. We have humoral, we have the CMI. So it's run by T cells. T cells, of course, respond by either interacting with the cells to kill them or producing cytokines. So the major role, of course, is to identify and any foreign invaders, and don't want to go into all those. Um, so the main functions of T cells will be to recognize peptide fragments and presented to them by antigen-presenting cells via T cell receptor. So that is one of the main roles, of course, of T cells, is that they are able to recognize antigens. Antigens are being presented to them in form of peptide fragments. Then, of course, uh, the adaptive immunity, the humoral, that is obvious. We know that. All right. So T cell subpopulations, we already talked about these subpopulations. We talked about T helper being Th1, Th2, Th17, and Tregs. We already talked about this, and we've seen the kind of cytokines they produce. We also talked about CD8. There are types of cell-mediated responses. We have delayed hypersensitivity, and we have direct cytotoxicity. So of course, delayed hypersensitivity is run by Th1 cells. Then direct cytotoxicity is mediated by T CD8 or T cells. And we say we have two types of CD8, TC1 and TC2. And TC1 behaves like Th1 and TC2 behaves like H2. Right. So I don't want to dwell on delayed hypersensitivity because we are going to talk about hypersensitivity in future. We have a whole topic on hypersensitivity. So we'll discuss hypersensitivity in details. There are four types of hypersensitivity. I think it's only one that is not cell mediated. The rest of the hypersensitivities are cell mediated. It's type 1, type 2, 3, type 4. Then CD8, we already talked about CD8 before. We said uh, antigens are usually presented via MHC1 when they're intracellular. And then, of course, they undergo apoptosis or lysis. They are killed using perforin and granzyme. And this is the same thing that happens in the case of natural killer cells. So CD8s are able to kill tumors. They're able to kill transplanted allogenic cells. They're able to kill tumor cells. They're able to kill virally infected cells because they have perforin and granzyme in them. Yeah, that is it. So I want to bring us to the end. As you've seen, cell-mediated immunity is just a repetition of what you've already talked about when you are discussing adaptive immunity in general. And so the of course, the main form of adaptive immunity is cell-mediated immunity. We've detailed so many things about it. I don't want to go back and repeating what we have already talked about. At this point, maybe I welcome any questions or clarifications before we call it an end. Before we end this class, maybe ask if you have any questions that you'd want responses to, you'd ask. And then I think we've discussed with your class rep tomorrow. We should be having another session. This session maybe will focus on humoral immunity. And probably get a bit of now the clinical immunology. Any questions, Victor?
So I would like a clarification. As we began, we said that the pathogens or the microorganisms, they have the PAMs, the pathogen-associated molecular patterns. So is there a possibility that these T-cell would also recognize the pumps rather than requiring the APC.
No, that's a good question. That's a very, very good question. All right. So, ideally, they do so. Technically, they are going to do so. They're going to recognize pumps because are referred as antigens. They are proteinaceous in nature. The only thing that we are talking about pumps in terms of innate immunity because these are conserved. I'm sure you are aware that mutations are happening in most of these pathogens. Mutations are happening. That is why they're able to evade some of the drugs, they're able to evade the immune system. But what the innate immune system does is that it looks for the conserved genes, the conserved antigens that are present, and that is what we are referring to as pumps. For instance, it is very difficult for bacteria to change its flagella. It will remain like that, okay? But the T cell is also able to recognize the flagella and know that this is a foreign protein and produce antibodies against it. So it's true, it's possible. But that is only possible if the innate immune system has been innate bacteria and now it has gotten to the adaptive immune system because it's sequential. The innate immune system has to come into play. Once it fails, then the adaptive comes into play, and antibodies are going to be generated against foreign proteins that the body is seeing as foreign, including those of flagella, including those of the peptidoglycans, including those of the lipopolysaccharides or double-stranded DNA or single-stranded RNA, CPGs, all those things that we talked about being pumps. So that's a very, very good question. Thank you. So that is possible. It happens, of course.